right, so to summarize what we talked about in the last lecture, here's sort of a summary, and this actually does have an error in it, so I want to correct it. So first of all, cells in a hypotonic solution. Well, let's talk about animal cells first. So an animal cell in a hypotonic solution, what's going to happen is water is going to enter the cell because the cell is going to be hypertonic to the solution. So we have hypo out here. We have hyper inside. Remember, water goes towards hypertonic, towards the side with less water and more solute. And this cell can actually burst, or sometimes it's called lyse, which means to explode. So the way that animal cells survive is that they have different coping mechanisms. In a multicellular organism like us, we keep our cells isotonic to our solution through things like hormones that control how much urine we make, by how much water we drink, and how much we sweat. And so these things all are taken into account, and our hormones keep our blood cells, even all of our cells actually, isotonic to the solution around them. Plant cells, hypotonic outside, they do not burst, and you need to know this for the test. What will happen is water will come in, but it will get stored in the central vacuole. And that will cause pressure. This will swell up really big and create pressure on this cell wall. And then the plant will actually look great. It will look turgid, which means stiff. This is when the plant will look healthy. Scenario number 2, isotonic. So what happens in an isotonic solution, remember, is that you have equal concentrations of solute. This is normal for an animal cell. Like a multicellular organism's animal cell, I should say. We have equal amounts of water going in both directions, so our cells stay the same. Now, what about in a plant cell if it's isotonic? This is the part where it's a mistake. The cell will actually not be normal. That is wrong. When your plants are isotonic, they're actually going to look a little bit wilted because the cell, the vacuole is not going to create heavy pressure on the cell wall. And so the cell's going to look a little bit wilted, even though water is going in and out, so it's not going to really shrivel up, but it's going to be slightly limp. This is not normal for a plant. This is normal for a plant. Hypotonic solution outside. Scenario 3, hypertonic solution. So you put your blood cells in salt water, and now it's hyper. Water is going to mostly be leaving the cell, and your cell will shrivel up. There is a name for it. It is called crenation, but you don't have to know that for a test. Now, in a plant cell, there is a name for it too. It's called plasmolysis, and this is where the plant shrivels up so much that the cell membrane collapses in, and it's not even pushing against the cell wall at all. You'll actually see empty space here. Typically, this will kill your plant. It will be really shriveled, and then it'll turn brown, and then it'll die. And this is in some real cells. This is a normal red blood cell. Red blood cells have a little dip in the middle like this. This is in a hypotonic solution, so water's going in, and this cell would eventually burst. And this is in a hypertonic solution, and the reason you're seeing these little spiky things is because this cell is actually shriveled up. This is a plant cell in a hypotonic solution on the left. The cell, you can see how the chloroplasts are smashed up against the cell membrane. You can't see the vacuole, but it's full of water, and it's creating pressure to make the cell stiff. And over here on the right, this is a plant cell in a hypertonic solution, and so the water has left the cells, and you see the gap here between the cell membrane and the cell wall, and this plant would look really, really wilted. It probably would die. All right, so how do organisms monitor their balance? Well, like I said, in animals, we use hormones to do that. But in little organisms like this organism, this is a Paramecium, and they do not actually have an isotonic solution around them. They live in fresh water, like in a lake. So the water around them is going to be hypotonic, and they are going to be hypertonic. Water tends to come in. But the Paramecium has something called a contractile vacuole. Looks kind of like a little flower. It swells up, and it squirts out excess water. And this prevents the Paramecium from exploding. All right, sorry, moving along past these. Penicillin, the way it kills bacteria is it actually punches holes in the cell wall, and then water comes flooding in, and the bacteria burst. Because they, with just a cell membrane and not a cell wall around them, they cannot control their water, prevent themselves from exploding. All right, so we've talked about passive transport and passive transport, diffusion, and osmosis. Neither of those require energy. Diffusion was when solutes moved high to low, and osmosis was when water moved high to low. Facilitated diffusion is just like simple diffusion. The difference is that instead of the solute going high to low through the phospholipid bilayer, it's going through special proteins in the membrane, because the cell membrane is not permeable to a lot of things, like sugar, for example, or sodium or potassium. How do those things get in if your cell needs them? Well, they cannot pass through the phospholipids, but if you have a protein carrier, you can. These proteins are called channels, and they're very specific. You have a channel just for glucose, a channel just for sodium. You have all different kinds of channels. Facilitate means to help. So these are channels that are helping move substances still from high to low concentrations only, but no energy is necessary. So imagine if this is the phospholipids, and then this is a protein in the membrane, and this is more phospholipids here. And this protein is a channel, it's like an opening. Maybe let's say this one is for glucose. Then what would happen is, if you have more glucose outside than inside, glucose will tend to flow through this membrane protein into the inside. So if you see a picture like this, where it's a protein in the membrane that's carrying something through from high to low concentrations, that's going to be called facilitated diffusion. But if you see the substance going directly through the membrane, like here, going through the phospholipids from high to low, that's going to be simple diffusion. So simple diffusion, facilitated diffusion. No energy necessary. You have hundreds of different protein channels. Glucose is an example of a molecule that's carried in by facilitated diffusion. So let's talk about that for a second. What actually is carried in and out by different methods? Well, first of all, oxygen, carbon dioxide, and anything that's lipid soluble or hydrophobic. So remember, oil and water don't mix. So anything that's lipid soluble that would dissolve in oil, those things can pass through the membrane very easily. Oxygen and carbon dioxide, steroids, cholesterol. Easily go from high to low concentrations straight through the phospholipids. Anything that's polar, has like slight charges, or things that have full charges, like ions, so like sodium, potassium, calcium. That should not have an A, sorry about that. Those things are not going to be able to go through the phospholipids. They are going to require a protein to carry them through by facilitated diffusion. Remember, it's only called facilitated diffusion if it's going through a protein from high to low concentrations, because things don't naturally go from low to high. Just like going downstream. If I drop a leaf into a stream, which way is it going to go? The direction the stream is flowing, from high to low. It's not going to go backwards unless I apply energy to it. So also water. Water does not flow into your cells super fast on its own, but there are proteins in the membrane called aquaporins that specifically carry water. And because of those aquaporins, which all your cells have, water pretty much flows through very quickly, as if it passed through the membrane on its own. So transport proteins, how do they make it selective? Because depending on which proteins you have and how many, that's going to determine what can and can't get in and out of a cell. So if a cell has more aquaporins, for example, it can transport more water. If a cell is missing a sodium protein to carry in sodium, then that cell will be impermeable to it. So every cell, depending on what proteins are in the membrane of that cell, that will determine what things can and can't pass. The phospholipids are pretty much identical in every cell. So these things can always pass. But the proteins are going to determine what polar things and what charged things can pass in and out of the cell. This is facilitated diffusion. So this is a membrane protein. This is a lower concentration of this item, and this is higher concentration, and the item flows through the protein, no energy necessary. This is called facilitated diffusion. If it was just going through here, it would just be simple diffusion. This is another picture. Sometimes the protein changes shape as the molecule goes through, so that's what they're showing in this diagram. But notice it's still going from high to low concentrations. All right, and that brings us to the last thing, which is active transport. So active transport is the opposite of everything we've talked about before. So everything we talked about before was called passive transport because no energy was required. So that was diffusion, osmosis, and facilitated diffusion. This active transport requires energy, ATP typically. This is going to go against the gradient. So imagine you're in a boat and the boat gets a hole in it. Water coming into your boat would be moving water from high to low, from where there's more water outside to inside your boat. That would not require energy. That would be like osmosis. But if you don't want your boat to sink, you're going to have to use energy, and you're going to have to get a bucket, and you're going to have to actively dump water out of your boat to keep your boat floating. That's like you are providing the ATP, the energy, to move it against the gradient. Or another example, you're in a boat and you want to go up a stream. If you want to go downstream, you don't have to do anything. You just sit there, your boat moves the way you want to go. But what if you want to go upstream against the flow? You're going to have to use energy. You're going to have to row your boat or get a motor for your boat or something. So sometimes the cell also needs to bring in a molecule against the concentration gradient. Bring it from where it's low to an area where it's already a little bit higher. Like, for example, if it needs a nutrient, the cell still needs it just because it's not in high concentrations outside. It still needs to bring it in. So it may have to pump things against the concentration gradient. For example, cells may actively transport calcium out. If it's active transport, the question is calcium more concentrated inside or outside? Well, if it's active transport and it's pumping out, then it must be higher concentrated out. In other words, if it was lower concentration out, so if you had less calciums out, then that would be the natural way it would go. But if you already have lots of calciums out here, your cell's not going to want to take them this way. The natural way for them to go would be high to low. So your cell would have to use energy, active transport, to pump the calcium out. This is showing active transport. Usually you'll see something like the word energy or ATP, and that's one big clue that what they're showing you in the picture is active transport. The other thing is they'll be showing it going from low to high. So there's only 2 here, but there's a whole bunch here, and it's going in this direction towards the higher concentration. That must be active transport. All right, last thing, a special kind of active transport to move large things is called endocytosis, to take it in, and exocytosis to take it out. So endocytosis is where the cell surrounds and engulfs it. You may remember endosymbiotic theory from the last chapter. So this requires energy because the cell physically has to move around and eat the particle. And then exocytosis is the opposite. Sometimes you have a vesicle, it travels to the membrane, it fuses with the membrane, and the products are exported. In both cases, it says here that everything gets packed in a vesicle. Those are the little, almost like a vacuole. And there's 3 specific endocytoses. So let me go through those really quick. Phagocytosis, the cell eats something really big, like a bacteria. This is like what your white blood cells do. And then they digest like bad stuff. Receptor-mediated endocytosis, this is where the cell has little receptors on the surface, and a molecule binds to that, and that sort of triggers the cell to engulf whatever that particle is. This is actually the way cholesterol is taken in. It said before it could pass through the membrane because it's lipid soluble, but actually your cell's typically taking cholesterol this way. A specific solute binds to the receptor, the cell eats it. And penocytosis is for when a cell surrounds and engulfs kind of like a liquid droplet with like a bunch of little particles. So instead of eating one giant thing, it's eating a bunch of little things. So phagocytosis, receptor-mediated endocytosis, and then pinocytosis. Those are the three. And again, this involves a vesicle forming after it takes it in.